Welcome to the deep dive. Today, we're really zeroing in on two absolutely critical areas, especially if you've got a prelims exam in medical sciences coming up: cancer and immunity.
Yeah, absolutely. These topics, they can feel, well, enormous, can't they?
Exactly. So our goal today is to pull out the core stuff,
Yeah.
the high-yield concepts, and try to make them as clear and sticky as possible.
Precisely. We've basically synthesized info from a whole range of key resources, your standard medical textbooks, some concise study guides, specific PDFs on cancer, on immunity,
Right.
to give you that foundational knowledge you really need. Think of this as maybe a strategic briefing, highlighting the key intel for your prep.
Okay, sounds good. Let's start trying to unpack cancer then. When you're looking at what seems like this endless list of tumor types, the naming system itself actually gives you some pretty helpful shortcuts.
It really does. It seems daunting at first, but there's logic there.
So benign tumors, for instance, they generally have that oma suffix, right? We see this in lipoma, that's a non-cancerous fat tissue growth.
Right, lipoma.
Glioma.
Glioma, yeah, usually a non-malignant brain cell tumor. And leiomyoma, that's benign smooth muscle. And chondroma for cartilage. It's kind of like cracking a code, isn't it?
It is, yeah. That oma is your first big clue for benign. Now, when we get to malignant tumors, the naming, it shifts. Depends on the tissue where the cancer started.
Okay.
So malignant tumors coming from epithelial tissues, the linings of organs, structures, those are carcinomas.
Carcinomas, got it.
Yeah, examples you'll definitely see are things like adenocarcinoma, often starting in gland tissue,
Yeah.
and basal cell carcinoma. That's a
Yeah.
very common type of skin cancer.
Right. And then there are sarcomas.
Exactly, sarcomas. Those are the malignancies that come from connective tissues, things like bone, cartilage, fat, muscle.
Ah, okay. So chondrosarcoma would be cartilage cancer.
Yep.
And osteosarcoma, bone cancer. Okay, recognizing those suffixes like carcinoma and sarcoma could save some real time on an exam. Absolutely. And it's also important to remember some cancers just have their own specific names, like lymphomas, those are cancers of lymphatic tissue.
Right.
And leukemias are cancers of the blood-forming cells. Another term you might bump into is carcinoma in situ, or CIS.
CIS. What's that exactly?
Well, interestingly, it describes malignant cells, but they're pre-invasive. They haven't yet broken through the basement membrane, that layer separating tissues.
Okay. So it's like cancer waiting to happen.
Kind of, yeah. For your prelims, understanding that CIS is a precursor stage, a pre-invasive stage, that's definitely key.
Okay. Let's move on to the fundamental characteristics of cancer cells. What makes them tick? We often talk about the hallmarks of cancer.
The hallmarks, yes. These are essentially the capabilities that cancer cells have to acquire to become, well, cancerous.
We probably don't have time for a super deep dive into all of them.
No, not exhaustively, but grasping the main ideas is crucial.
So things like sustained proliferative signaling, they're basically stuck on grow, right?
Exactly. Constantly telling themselves to divide. And evading growth suppressors, they just ignore the normal stop signals.
And genomic instability, their DNA gets all messed up.
Yeah, and that genomic instability is a really, really important point. It fuels more mutations, more changes.
Makes them harder to treat, I guess.
Definitely complicates treatment strategies because the tumor cells become so varied. Other hallmarks include enabling replication immortality. They basically become immortal. They can divide endlessly, often by messing with their telomeres, which we can touch on.
Okay.
Reprogramming energy metabolism, they change how they make energy to fuel all that rapid growth. And resisting apoptosis, they get very good at dodging programmed cell death.
So what does this actually look like at the cell level, this transformation?
Well, the cells essentially go rogue. They show anchorage independence, meaning they don't need to be attached to anything to grow, unlike most normal cells.
Right, they can just float around and still multiply.
Yep. They're immortal, as we said. They also show anaplasia, meaning they lose their specialized features, they become less differentiated.
Less like the original cell type.
Exactly. And plemorphism, the cells themselves become varied in size and shape. And then you have cancer stem cells.
Oh, stem cells, those sound problematic.
They really are. Because they can self-renew, like normal stem cells, but they can also churn out various types of cancer cells within the tumor. This means they can potentially drive recurrence even after treatment seems successful.
Okay, that's a key concept. What about tumor markers? That seems like something important for exams.
Definitely. Tumor markers are substances, could be hormones, enzymes, genes, antigens, even antibodies produced by cancer cells or sometimes by the body in response to cancer. You can often detect them in blood, urine, or other bodily fluids.
So they're useful for diagnoses, monitoring.
They can be useful for all of that risk assessment, sometimes diagnosis, and definitely for monitoring how well treatment is working or checking for recurrence. But, and this is vital to know, elevated levels aren't always proof of cancer.
Right. Other things can cause them to go up.
Exactly. Non-cancerous conditions can also raise marker levels, so you need to understand their utility, but also their limitations. Concepts like sensitivity and specificity are key here. How good the test is at identifying people with the disease and people without it.
Makes sense. Okay, so how do these cancerous changes actually start? You hear cancer is a disease of aging.
Largely, yes. It's often driven by what we call clonal proliferation. Imagine one cell gets a mutation, giving it a slight growth advantage.
And it starts outgrowing its neighbors.
Precisely. And here's a really crucial point, especially for prelims. It usually takes multiple mutations accumulating over time for a cell to become fully cancerous, not just one hit.
Multiple hits needed. Got it.
Yeah.
What about epigenetics?
Ah, yes. Epigenetics is huge, too. These are modifications to the DNA or to the proteins DNA wraps around that change how genes are expressed without actually altering the DNA sequence itself.
We like switching genes on or off inappropriately.
Exactly. And these epigenetic changes can play a really significant role in driving cancer development as well. It's not just about the DNA code changing.
Okay. So connecting this back, these mutations or epigenetic changes often mess with genes controlling cell growth signals, right?
Absolutely. For instance, cancer cells might start making their own growth factors. That's called autocrine stimulation. Or they might develop way more receptors for growth factors, making them super sensitive to even tiny signals.
Or the signals inside the cell get stuck on.
Yes. Like mutations in the RAS protein, that's a classic example, it gets stuck in the on position. And, crucially, the brakes can fail. Tumor suppressor genes like RB and TP53, which normally stop uncontrolled growth, can be inactivated.
So the go signals are stuck on and the stop signals are broken.
A dangerous combination. Plus, pathways that normally trigger apoptosis program cell death can be shut down, letting these damaged cells survive when they really shouldn't.
Let's dig into oncogenes and tumor suppressor genes a bit more. These sound like really high-yield concepts for exams.
Definitely are. Oncogenes are basically mutated forms of normal genes called proto-oncogenes.
Proto-oncogenes. Those are the normal go signals.
Yeah, they're involved in normal cell growth and division, but when they mutate or get overexpressed, they become oncogenes and push the cell towards cancer, like a stuck accelerator pedal.
Okay. And tumor suppressor genes are the brakes.
Exactly. They normally inhibit cell proliferation, repair DNA damage, initiate apoptosis if needed. They're the body's natural safeguards.
So how do proto-oncogenes turn into oncogenes? What are the mechanisms?
Good question. Several ways. You can have point mutations, tiny changes in the DNA code, or chromosome translocations where bits of chromosomes break off and swap places.
Right?
That can stick a proto-oncogene next to a strong promoter or fuse it with another gene to create a hyperactive protein. A classic example you might need to know is N-mic amplification in neuroblastoma.
Amplification.
Yeah, gene amplification. That's where the cell makes multiple copies of the proto-oncogene, leading to way too much of the growth-promoting protein being made.
Okay. Point mutations, translocations, amplification. Got it. And for tumor suppressor genes, how are they lost?
Well, if they get mutated or deleted, that critical regulation, that braking system, is gone. We often talk about loss of heterozygosity, or LOH.
LOH?
Yeah, you normally have 2 copies of each gene. LOH is when you lose the one remaining functional copy of a tumor suppressor gene, leaving you with none. Gene silencing is another way that's often through epigenetic mechanisms, basically switching the gene off without mutating it.
And caretaker genes, what do they do?
Caretaker genes are a type of tumor suppressor genes specifically involved in DNA repair. If they malfunction, the cell can't fix DNA damage properly.
Leading to more mutations.
Exactly. It leads to genomic instability, making the cell much more likely to accumulate those other cancer-promoting mutations in oncogenes and other tumor suppressors.
Okay, this interconnectedness is complex but crucial. What about angiogenesis? That always comes up.
Absolutely critical concept. Angiogenesis, the formation of new blood vessels. Tumors, once they get beyond a tiny size, they need their own blood supply.
For nutrients and oxygen, right?
Nutrients, oxygen, and also a route for waste removal and, unfortunately, for metastasis. Factors like TP53, the tumor suppressor, can actually inhibit angiogenesis. But tumors often activate pro-angiogenic factors, signals that say, build more vessels here.
And they're inhibitors, too.
Yes, the body has natural angiogenesis inhibitors, and tumors often find ways to suppress those. So understanding this balance, pro versus anti-angiogenic factors, is key. And remember telomeres.
The caps on the ends of chromosomes, they shorten with each division?
Normally, yes. It acts like a cellular clock, limiting the number of divisions. But many cancer cells activate an enzyme called telomerase. Right? Rebuilds the telomeres. This allows them to bypass that normal limit and achieve replicative immortality. Keep dividing forever.
Wow, okay. So genetics clearly plays a huge role. How does inheritance fit in?
We need to distinguish somatic versus germline mutations. Somatic mutations happen in individual body cells during a person's life. They're not passed on. Germline mutations occur in the sperm or egg cells. These can be inherited, passed down to offspring. This explains why you see some families with a much higher predisposition to certain cancers.
Right, like BRCA mutations and breast cancer risk.
Exactly. And for prelims, don't forget the link between chronic inflammation and cancer.
Inflammation. How does that promote cancer?
Well, chronic inflammation, think long-term irritation or infection, involves the constant release of cytokines and free radicals by immune cells. These can damage DNA directly, promote cell proliferation, and create a microenvironment that actually helps tumors grow and spread.
Like the H. pylori example.
The perfect example. Chronic Helicobacter pylori infection is a major risk factor for stomach cancer and a type of lymphoma called multilymphoma. The inflammation itself paves the way.
And viruses. You mentioned those earlier, too.
Yes, definitely know the main cancer-associated viruses. Hepatitis B and C for liver cancer, Epstein-Barr virus linked to Burkitt lymphoma and nasopharyngeal carcinoma, Kaposi sarcoma herpes virus, HPV, human papillomavirus causing cervical and other cancers.
Right, HPV is a big one.
And HTLV, human T lymphotropic virus, linked to a type of leukemia lymphoma. These viruses can mess with cell cycle control genes, introduce oncogenes, or cause chronic inflammation.
Okay, so once cancer starts, how does it spread locally? Invasion.
Yeah. Local spread or invasion is the first step. Cancer cells multiply, creating mechanical pressure. They also secrete hemolytic enzymes.
Enzymes that chew through tissues.
Exactly. They degrade the extracellular matrix, the scaffolding holding tissues together. They often lose cell adhesion molecules, the glue that normally keeps cells in place, making them less sticky.
So they can detach more easily.
Right. And they become more motile, able to move. Eventually, they can push into nearby tissues and even penetrate into blood or lymphatic vessels. That's called intravasation.
Intravasation, getting into the vessel.
Yes.
Yeah.
And later, extravasation is getting out at a distant site.
Is there a model for how invasion happens?
There's a useful framework called the three-step theory of invasion. It's good to remember for exams. Step 1. Tumor cell attachment to the matrix, often using molecules like fibronectin and laminin.
Right, attachment.
Step 2. Degradation of that matrix by those enzymes we mentioned.
Degradation.
And step 3. Locomotion. The cell actively moves through the degraded matrix, often using little projections called invetopodia.
Invetopodia, okay. Attachment, degradation, locomotion. And that leads to metastasis.
Invasion sets the stage for metastasis, yes. Metastasis is the spread to distant sites, and it's what makes cancer so deadly, usually. It can happen through direct extension into neighboring organs or seeding into body cavities.
Like the abdominal cavity.
Exactly. Or, most commonly, via transport through the blood or lymphatic systems after intravasation. Then the cells have to survive the journey, extravasate, get out of the vessel, and establish a new tumor, a secondary tumor, at the distant site. It's a complex and inefficient process, actually, but devastating when it succeeds.
And we use staging to describe how far it spread.
Yes, staging is crucial for prognosis and treatment planning. The T&M system is key for prelims. T for tumor size and extent.
E for tumor.
N for node involvement, whether regional lymph nodes are affected.
N for nose.
And M for metastasis, whether distant metastases are present.
TNM. Okay.
And more generally, you'll hear clinical stages 1 to 4. Stage 1 usually means it's confined, localized.
Stage 1 confined.
Stage 2 indicates local invasion, maybe slightly larger or deeper.
Stage 2, local invasion.
Stage 3 typically means spread to regional structures, like nearby lymph nodes.
Stage 3, regional spread.
And stage 4 means it's metastasized to distant sites, distant spread.
Stage 4, distant metastasis. Okay, that makes sense. Clinically, how does cancer show up? What are the common symptoms?
It varies hugely, but some common manifestations include pain, This can be from the tumor pressing on nerves or organs, blocking things, or directly invading tissues.
Right. Pain makes sense.
Fatigue is another huge one, really common, often debilitating. It could be due to sleep issues, biochemical changes from the cancer or the immune response, anemia, lots of factors.
And cachexia. That sounds serious.
It is. Cachexia is severe malnutrition, profound weight loss, muscle wasting, anorexia. It's not just from not eating enough. The body's metabolism is altered.
Wow. Anemia seems common, too.
Very. Can be from chronic bleeding, like from a tumor in the gut, poor nutrition, side effects of therapy, or the cancer actually getting into the bone marrow and disrupting red blood cell production.
And if it affects the marrow, that impacts white cells and platelets, too.
Exactly. You can get leukopenia, low white blood cells, increasing infection risk, and thrombocytopenia, low platelets, increasing bleeding risk, often due to marrow invasion or, very commonly, chemotherapy.
So infection risk goes up.
Definitely, especially if neutrophil counts drop. And lastly, keep paraneoplastic syndromes in mind.
Paraneoplastic, what does that mean?
These are weird signs and symptoms that are not directly caused by the tumor mass itself, its spread, or hormones you'd expect from that tissue type.
So indirect effects.
Yeah, often caused by substances the tumor releases unexpectedly, like hormones or cytokines, or sometimes by an immune response against the tumor that cross-reacts with normal tissues. They can be quite diverse and sometimes are the first sign of cancer.
Fascinating. Okay, what about treatments? Chemotherapy.
Chemo uses drugs to kill rapidly dividing cells. Key thing for prelims. It can be used in different settings. Induction therapy aims for remission, often in leukemias. Neoadjuvant is chemo before surgery to shrink the tumor.
Neo before. Makes sense.
And adjuvant is chemo after surgery to mop up any leftover cells.
Adjuvant after. Got it.
And remember, cells aren't equally sensitive all the time. Sensitivity depends on where they are in the cell cycle. Chemo often targets cells actively dividing.
What about radiation?
Ionizing radiation works by damaging the DNA of cancer cells, hopefully beyond repair. A key point is cumulative lifetime dose limits. You can only give so much radiation to one area over a lifetime to spare healthy tissues.
Right, limits.
And there's brachytherapy, that's internal radiation, where radioactive sources, like little seeds, are placed directly inside or very close to the tumor.
Brachytherapy, internal.
Okay.
And surgery.
Surgery is still fundamental. For biopsy to get a diagnosis, for lymph node sampling, often using sentinel node biopsy to check the first few nodes the tumor drains to.
Sentinel node?
And, of course, for removing the tumor itself. Achieving clear surgical margins, meaning no cancer cells are found at the edge of the removed tissue, is really critical to reduce recurrence risk. Careful handling is also key to avoid spilling tumor cells.
And finally, immunotherapy. That seems like a huge area now.
It really is a major pillar of cancer treatment. The basic idea is to boost the patient's own immune system to recognize and fight the cancer.
So using the body's own defenses.
Exactly. There are various ways giving cytokines like interferon, using monoclonal antibodies to target cancer cells directly, or, very excitingly, using checkpoint inhibitors.
Checkpoint inhibitors.
Yeah. These drugs basically release the natural brakes on the immune system, allowing immune cells, like T cells, to attack the cancer more effectively. Understanding that basic principle, unleashing the immune system against cancer, is key.
Okay, wow. That's a lot on adult cancers.
Yeah.
What about cancer in children? You mentioned there are differences.
Yes, critical differences, very important for prelims.
Yeah.
The most common types in kids are leukemias, especially, acute lymphoblastic leukemia, OLL,
Well, okay.
and tumors of the brain and central nervous system. Interestingly, CNS tumors are also the most common solid tumors in children.
CNS tumors are big in kids then.
Yes. We also see sarcomas, cancers of connective tissue, and embryonic tumors. These start during fetal development and often have names ending in soplast.
Like neuroblastoma.
Neuroblastoma, retinoblastoma, Wilms tumor, kidney, hepatoblastoma, liver. That fat blast suffix is a clue for these embryonic origins.
And where do most childhood cancers originate from, embryologically?
Many arise from the mesodermal germ layer. That's the layer giving rise to connective tissues, bone, muscle, blood, kidneys, lymphatics. This is different from adults where most cancers are epithelial carcinomas.
Mesodermal origin.
Ah.
Okay. Any other characteristics?
They're often diagnosed during growth spurts. They tend to grow very fast, and unfortunately, they've often already metastasized by the time of diagnosis.
Interesting.
Also, statistically, boys are slightly more affected than girls.
What about teens and young adults, say 1539?
Yeah, adolescents and young adults, or AYAs, they actually have a higher overall malignancy rate than younger kids. The common cancers shift a bit too. You see more Hodgkin lymphoma, leukemia still, germ cell tumors like testicular cancer, CNS tumors still, non-Hodgkin lymphoma, thyroid cancer, sarcomas.
So a broader mix.
Yes, and even some cancers more typical of older adults start appearing, like breast, cervical, melanoma, some GI cancers. It's a transitional age group.
What causes childhood cancers? Is it genetics?
Environment. It's usually multifactorial, a complex interplay. Genetic predispositions definitely play a role on genes, tumor suppressor gene mutations, sometimes specific chromosome abnormalities like aneuploidy or translocations.
Like Down syndrome.
Exactly. That's a crucial link for exams. Children with trisomy 21 Down syndrome have a significantly increased risk of leukemia. That's probably the most common known genetic abnormality linked to childhood acute leukemia.
Good one to remember.
Any others? Yes, there are familial cancer syndromes linked to specific childhood cancers, like hereditary retinoblastoma or syndromes predisposing to Wilms tumor.
Okay. What about environmental factors?
They seem to play less of a role than in adult cancers, but some links exist. Prenatal exposures, like to the drug DES years ago, or maybe certain pesticides. Childhood exposures, like certain drugs, definitely ionizing radiation. And some viruses, like Epstein-Barr, linked to Burkitt lymphoma and HIV.
Got it. How's the prognosis generally?
Thankfully, it's improved dramatically. The overall cure rate for childhood cancer is now around 80%.
That's great news.
Huge progress. Mortality rates are dropping, partly due to better treatments developed through clinical trials, which most children with cancer participate in. There's a big focus now on targeted therapies with fewer long-term side effects and research into understanding those late effects of treatment.
Okay, that's a comprehensive overview of cancer. Let's shift gears now to immunity, our body's defense system.
Right, another huge topic. We usually think of it in 3 lines of defense.
3 lines. Okay, what's the first?
The first line is all about barriers. Physical barriers, skin, mucous membranes lining your airways, and gut. Mechanical things like coughing, sneezing, cells sloughing off.
Keeps things out physically.
Exactly. And biochemical barriers, too. Secretions like saliva, tears, mucus contain antimicrobial substances. Think lysozyme. Also, antimicrobial peptides like cathelicidins and defensins.
And our normal bacteria.
Yes, the normal flora, like lactobacillus in the vagina or gut bacteria. They compete with pathogens for space and nutrients, and some even produce substances that inhibit invaders. They're part of that first line.
Okay, first line. Barriers, physical, chemical, microbial. What's the second line?
Second line is innate immunity. This kicks in if the first line is breached. It's rapid, non-specific, meaning it reacts similarly to different threats, and it's the first immune responder.
Innate, non-specific, fast. What does it involve?
A crucial process is phagocytosis, specialized cells engulfing and destroying pathogens or debris.
Eating the invaders.
Pretty much. It involves steps like opsonization, tagging the target to make it easier to eat.
Like putting handles on it.
Yeah, good analogy. Then recognition, adherence, engulfment into a vesicle called a phagosome, fusion with lysosomes full of digestive enzymes, and boom, destruction.
Who are the main phagocytes?
Key players you need to know. Neutrophils, also called PMNs. They're usually the first immune cells to arrive at an infection site, great at eating bacteria and debris, but they're short-lived. They form the main component of pus.
Neutrophils, first responders. Pus?
Then monocytes, macrophages. Monocytes circulate in the blood, but when they migrate into tissues, they mature into macrophages. They arrive a bit later than neutrophils, but are bigger, live longer, and are more versatile. They also help activate adaptive immunity.
So monocytes become macrophages in tissues.
Right, and there's this whole network, sometimes called the reticuloendothelial system or mononuclear phagocyte system, circulating monocytes, mobile macrophages, fixed macrophages in specific tissues, like Kupffer cells in the liver, alveolar macrophages in the lungs, histiocytes in the skin. They're stationed everywhere.
Okay. Any other innate cells?
Yep. Eosinophils weakly phagocytic but key players against parasites and in modulating allergic inflammation. Basophils, similar to mast cells, release histamine and other inflammatory mediators.
Basophils and mast cells involved in allergies.
Heavily involved, yes,
Yeah.
especially through IgE antibodies. Natural killer NK cells are really important too. They can kill virus-infected cells and some cancer cells without needing prior activation like the adaptive system.
NK cells, innate cancer killers.
In a way, yes. And even platelets get involved, releasing factors and interacting with coagulation.
Don't forget mast cells themselves.
Absolutely. Mast cells reside in connective tissues, acting like sentinels. When activated by injury, chemicals, immune signals like IgE, or pathogen patterns via toll-like receptors, TLRs, they degranulate, releasing potent stuff like histamine and proteases immediately. They also synthesize lipid mediators like prostaglandins later, big drivers of inflammation.
Okay. And cytokines, those are the messengers, right?
Crucial signaling molecules, yes, lots of them. Interleukins, ILs, are a big family like IL-1, which is pro-inflammatory, and IL-10, which is anti-inflammatory. They regulate immune cell communication.
Pro and anti-inflammatory interleukins.
Interferons, IFNs, are vital against viruses. Type I interferons, alpha and beta, make cells resistant to viral infection. Interferon gamma activates macrophages, making them better killers.
Interferons, antiviral, macrophage activation.
And tumor necrosis factor alpha, TNFC. Mainly from macrophages. It's a major pro-inflammatory cytokine, causes fever, boosts other inflammatory proteins, recruits white blood cells. But too much can cause problems like cachexia or septic shock.
TNF-alpha.
Yeah.
Inflammation, fever, recruitment. Okay. What about the visible signs of inflammation? Redness, swelling.
That's the vascular response. Initially, brief vasoconstriction, then vasodilation, more blood flow, causing redness and heat. Then increased vascular permeability, leaky blood vessels. Fluid and plasma proteins leak out into the tissues, causing swelling, edema, and sometimes pain. This leakage is called exudation.
Exudate. Can it look different?
Yes. It can be serious, watery, like a blister. Fibrinous, rich in fibrinogen, sticky, purulent. Pus contains neutrophils, dead cells, bacteria, or hemorrhagic contains red blood cells indicating vessel damage.
Got it. And systemically, fever.
Yes, systemic effects include fever pyrogens, fever-inducing substances like IL-1 and TNF-alpha, act on the hypothalamus to reset the body's thermostat. Leukocytosis, an increase in the total white blood cell count. Often with a left shift, meaning more immature neutrophils are being released from the bone marrow, a sign of active infection.
Left shift equals more immature neutrophils.
And increased synthesis of acute phase proteins by the liver, things like C-reactive proteins, CRP, and fibrinogen. You measure these in blood tests as markers of inflammation.
CRP, fibrinogen. Okay. What happens if inflammation doesn't resolve? Chronic inflammation.
Right. Chronic inflammation is prolonged, lasting weeks, months, or even years. It can happen if the acute response fails, or if there's a persistent irritant, like asbestos, or a microbe that macrophages can't easily kill, like tuberculosis bacteria.
What does it look like histologically?
Often characterized by the simultaneous presence of ongoing injury, inflammation, with lymphocytes and macrophages being prominent, and attempts at repair. A hallmark feature can be granuloma formation.
Granuloma.
Yeah, organized collections of activated macrophages, sometimes fusing into giant cells or forming epithelioid cells, surrounded by lymphocytes. It's the immune system's attempt to wall off something it can't eliminate. Think TB.
Okay, so what are the overall goals of inflammation, acute or chronic?
Fundamentally, limit and control the damage or infection, prevent it from spreading, control bleeding through clotting, initiate the adaptive immune response if needed, and ultimately prepare the area for healing and repair.
Healing? How does that work?
Healing involves two main phases after the initial inflammation. The reconstructive phase involves fibroblast proliferation. These cells lay down collagen for structural support. Epithelialization occurs, covering the wound surface, and myofibroblasts help contract the wound, pulling the edges together.
Collagen synthesis, covering, contracting.
Then comes the maturation phase, which can last a long time. The new tissue differentiates further, and the scar tissue gets remodeled and strengthened.
Is it always a scar?
Ideally, you get resolution returned to normal structure and function, but often, especially with significant damage, you get repair, which involves scar tissue formation. Debridement, removing dead tissue and debris, is often needed for proper healing.
And healing can go wrong.
Oh, absolutely. Dysfunctional healing can lead to lots of problems. Hemorrhage, forming adhesions, scar tissue bands connecting organs, infection, excessive scarring like keloids or hypertrophic scars, weak scars due to impaired collagen synthesis, problems with epithelialization or excessive contractures that limit movement.
Okay. That covers the first two lines. What about the third line? Adaptive immunity.
Right, the third line, adaptive or acquired immunity. This system works closely with innate immunity, but has key differences. It's specific, meaning it targets particular invaders, and it has memory.
Specificity and memory, key features.
The main players are lymphocytes, T cells, and B cells, and the antibodies produced by B cells, which is called humoral immunity.
And they respond to antigens.
Exactly. Antigens are molecules, usually on the surface of pathogens or foreign substances, that elicit an adaptive immune response. They're typically proteins or large polysaccharides, and they have specific regions called epitopes that antibodies or T-cell receptors bind to. Usually need to be fairly large molecules.
Where do lymphocytes develop?
They originate in the bone marrow. B cells mature primarily in the bone marrow after birth. It's a fetal liver before that. T cells migrate to the thymus for maturation.
T for thymus.
Right. In these primary lymphoid organs, lymphocytes undergo development, including processes to ensure they don't react against self-antigens, clonal deletion. Then they circulate and populate secondary lymphoid tissues, lymph nodes, spleen, tonsils, Peyer's patches in the gut, where they're likely to encounter antigens. Clonal development ensures each lymphocyte recognizes a specific antigen.
How do they get activated?
It usually starts with antigen-presenting cells, APCs, like macrophages or dendritic cells. They engulf the pathogen, break it down, and present fragments antigens on their surface to helper T cells, CD4 plus T cells.
APCs present antigen to helper T cells.
Yes, and the APC often secretes cytokines like IL-1 to help activate the T cell. The activated helper T cell then produces its own cytokines, which are crucial for activating B cells and other T cells, like cytotoxic T cells, to proliferate and differentiate.
So helper T cells are central coordinators.
Absolutely. Meanwhile, B cells can often recognize intact antigens directly through their B cell receptors, which are essentially surface-bound antibodies. T cells, however, only recognize processed antigen fragments presented by APCs on special molecules called MHC proteins.
MHC proteins. Tell me more about those.
Major histocompatibility complex proteins, crucial for T cell recognition. There are two main types you need to know. MHC class I is found on almost all nucleated cells. It presents internal antigens like pieces of viruses infecting the cell, or abnormal proteins from a cancer cell to CD8 plus cytotoxic T cells.
MHCI, internal antigens, 9 CD8 T cells.
Exactly. MHC class II is found mainly on APCs, macrophages, dendritic cells, B cells. It presents external antigens from pathogens that have been engulfed and broken down to CD4 plus helper T cells.
MHC2, external antigens, mana CD4 T cells.
Got it. So back to B cells. How do they make antibodies?
Once activated, usually with help from CD4 T cells, B cells proliferate rapidly and differentiate into plasma cells. Plasma cells are essentially antibody factories, secreting huge amounts of antibodies specific for the antigen that triggered the response.
Plasma cells equals antibody factories.
Some activated B cells also become memory B cells. These stick around for a long time, ready to mount a much faster and stronger response if you encounter the same antigen again. That's the basis of immunological memory and vaccination.
Memory B cells for secondary response. Makes sense. What about antibodies themselves? What do they do?
Antibodies, also called immunoglobulins, IgG, are proteins. Their structure allows them to bind very specifically to antigens. They're typically at least bivalent, meaning one antibody molecule can bind to at least two antigen molecules.
Different types of antibodies. IgG, IgM. Yes, there are different classes or isotypes with different functions. IgM is usually the first one produced in an immune response. Ig is the most abundant in blood, crosses the placenta, and is involved in many functions like opsonization and complement activation. IgA is crucial for mucosal immunity, found in secretions like saliva, tears, gut mucus. IgD's function is less clear, mostly found on B cell surfaces. And Ig is famous for its role in allergy and defense against parasites, working with mast cells and eosinophils.
IgM first, IgG most abundant, IgA mucosal, IgE allergy parasites. Okay. How do they fight pathogens?
Several ways. Agglutination clumping pathogens together, precipitation making soluble antigens insoluble, neutralization blocking the harmful effects of toxins or preventing viruses from entering cells, activating the complement system, which leads to lysis bursting of pathogens, and opsonization coating pathogens to make them more easily phagocytosed.
Lots of mechanisms. Now T cells again. We mentioned helper and cytotoxic T cells.
Right. T-cell activation also involves recognizing antigen presented on MHC molecules by APCs. This leads to clonal expansion, making many copies of the specific T-cells needed, and differentiation into effector cells and memory T-cells.
Memory T-cells, too.
Yep, just like B-cells for long-term immunity.
So helper T-cells, CD4+. What's their main job again?
They're the regulators, the conductors of the immune orchestra. They release cytokines like IL-2, IL-4, IL-5 that influence virtually all other immune cells. They help activate B cells to make antibodies, help activate CD8 T cells to become effective killers, and activate macrophages to destroy intracellular pathogens more effectively.
Okay, CD4 helpers are crucial regulators, and cytotoxic T cells, CD8+.
These are the direct killers. They recognize cells displaying foreign antigens on MHC class, I think virus-infected cells, cancer cells with abnormal proteins, or cells from a foreign transplant.
And they kill them.
Yes, they induce apoptosis, programmed cell death in the target cell. Very precise and effective.
Okay. How does the body avoid attacking itself? Tolerance?
Immunologic tolerance is essential. During lymphocyte development in the thymus for T cells and bone marrow for B cells, cells that react strongly against self-antigens are normally eliminated, clonal deletion or inactivated anergy.
So self-reactive cells get weeded out.
Mostly, yes. When this process fails, it can lead to autoimmune diseases, where the immune system mistakenly attacks the body's own tissues.
Makes sense. And immunization, active versus passive.
Immunization uses adaptive immunity's memory feature. Active immunization involves introducing antigens from killed or weakened microbes or just parts of them to stimulate your own immune system to produce antibodies and memory cells, gives long-lasting protection.
Like vaccines.
Exactly. Passive immunization is giving someone preformed antibodies or immune cells from another source. Think antibodies from mother to baby via placenta milk, or antibody injections for things like tetanus or rabies exposure. It provides immediate but temporary protection because the recipient's own immune system wasn't stimulated.
Active, long-lasting, passive, temporary.
Got it.
And allergy.
Hypersensitive. Allergy or hypersensitivity is an exaggerated or inappropriate immune response to normally harmless environmental antigens, allergens. There are different types, but two key ones for prelims might be type I and type 5.
Type I.
Type I is the immediate type, mediated by IgE antibodies bound to mast cells. When the allergen binds to IgE, mast cells degranulate instantly, releasing histamine and other mediators causing classic allergy symptoms, hives, hay fever, asthma, potentially anaphylaxis.
IgE and mast cells.
Okay, type IV.
Type IV is delayed type hypersensitivity, mediated by T cells, not antibodies. It takes longer to develop, 24-72 hours. Examples include contact dermatitis, like poison ivy, or the reaction to the TB skin test. Involves T cells recruiting and activating macrophages.
T-cell mediated, delayed. Okay, wow, that's immunity in a nutshell. How do we tie all this back to cancer for prelims prep?
Okay, connecting the dots. It's vital to see the interplay. Innate immunity plays a role. NK cells can kill some tumor cells directly, macrophages can too, and they also act as APCs to kickstart adaptive immunity against the tumor.
Bridging innate and adaptive. Exactly. Adaptive immunity is key for long-term control and immunotherapy. Cytotoxic T cells, the CD8+, recognizing tumor antigens presented on MHC-I are crucial killers. Helper T cells, CD4+, orchestrate the anti-tumor response. Antibodies can target tumor cells for destruction, too.
So the immune system can fight cancer.
It can, and does. It's called immune surveillance. But cancer cells evolve ways to evade it. Immunotherapy aims to overcome that evasion. Remember the genetic basis of cancer: oncogenes, tumor suppressor genes. These mutations create the abnormal proteins, tumor antigens, that the immune system can potentially recognize.
And inflammation's dual role.
Right. Chronic inflammation can promote cancer initiation and progression. But acute inflammation can be part of the anti-tumor immune response, recruiting immune cells. It's complex. Remember specific links like Down syndrome and leukemia risk for childhood cancer questions.
Okay. What else should someone focus on for exams?
Think about how hypersensitivity might show up maybe as a reaction to a drug used in treatment, or even rarely as a paraneoplastic syndrome. Know the main immune cell types and their basic anti-cancer functions. NK cells, CD8 T cells, macrophages, antibodies.
Tumor markers, again, useful, but with caveats.
Exactly. Know their potential use in diagnosis monitoring, but be very aware of sensitivity specificity limitations. And staging, TNM, stages 1 to 4, is fundamental for understanding prognosis and treatment approach.
Any tips for memorizing?
Definitely use mnemonics if they help you. Simple diagrams linking concepts, like draw the pathway from a proto-oncogene mutation to uncontrolled growth. For naming, really drill the suffixes. Oma, mostly benign. Carcinoma, epithelial malignant. Sarcoma, connective tissue malignant. For immunity, maybe a simple chart for EG classes or MHCI versus 2.
Focus on the common childhood cancers, ALL, CNS tumors, the blastomas.
Yes, and their mesodermal origin generally. Understand the core mechanism. Oncogenes are accelerators, tumor suppressors are brakes, and mutations mess them up.
Okay, so to wrap up,
Yeah, bringing it all together, we've hit a lot today. Cancer classification, how it starts, genetics, environment, epigenetics, how it spreads, how we treat it. Focusing on those key hallmarks and gene types. Then the immune system's layers, innate barriers, rapid innate response with phagocytes and inflammation, and the specific memory-based adaptive response with T cells, B cells, antibodies.
and we tried to highlight the connections, especially for prelims, immune surveillance of cancer, immunotherapy basics, the unique aspects of childhood cancers, high-yield concepts like staging and tumor markers.
Right. The goal was really clarity and giving you a memorable framework for these pretty intricate topics, breaking them down into manageable chunks.
Definitely helpful. So maybe a final thought for everyone listening as they continue their prep.
Well, maybe reflect on this incredible potential harnessing the body's own, incredibly sophisticated defense systems to actually fight cancer. Immunotherapy is built on that idea. Think about how far we've come and maybe dig a bit deeper into any part of this discussion that really sparked your interest or felt particularly challenging for your studies.
Great advice. Best of luck to everyone with their prelims preparation.